Good morning, student. In today's lecture, we are going to discuss anther and pollen culture. In that, we are going to see what is the introduction of the anther and pollen culture, then principle and factor affecting. First, we will see what is the anther and pollen. So, in this image, you can see this is flower, which is reproductive organ of the plant. So you can see the flower has sepals, petals, then this is anther, then it has a filament, then you can see this is ovary. So what we will see which is anther. So this is anther, right, which is bilobed, and inside this anther there are presence of microspore. This microspore present in the pollen sac. So there are 4 pollen sac or 2 chambers where the microspores are present. So now we will see what is anther culture. So for this anther culture, or to initiate the anther culture, you are going to use explant as an unopened flower bud. Then you can collect this unopened flower bud from the greenhouse or from the field. You can collect that unopened flower bud in the polythene bags. Then you can take it into the lab. Then in aseptic condition, you are going to excise the anther at precise and critical stage. Keep in mind the anther, generally the anther which contains the microspore at first mitotic stage, those are selected. How you can select? You can select one anther, then press that anther, so that what happens, the microspore or the pollen grain which are present inside the anther, that will come out. Then stain that pollen grain with the acetocarmine, and then you will come to know the stage of that pollen grain, and according to that you can select the anthers. Then the excised anther you can culture on the nutrient medium, where that microspore which are present in the anther, that can give rise to callus tissue or the embryoid, and that callus tissue will develop into or they may give rise to organs or embryo, and that and ultimately that will develop into haploid plantlets. So keep in mind, from the anther culture you are going to produce haploid plantlets. The plantlets which are generated from the anther cultures are not diploid. So this is the one important application of anther culture, production of haploid plants. So now you can define the anther culture. Anther culture is a technique by which the developing anthers at precise and critical stage are excised aseptically from unopened flower bud, where they are cultured on nutrient medium, so that the microspores within the cultured anther will develop into callus tissue or embryoids that will give rise to haploid plantlets, either through organogenesis or embryogenesis. So you can see these are the photographs of previous batch practical. So you can see this is the petri plate containing MS medium, and that MS medium, the anthers are inoculated. You can see the anthers are intact without filament. So you are going to culture only the anther, not the filament. And in this way you can see the uninucleate pollen grain which are present in the anthers. Those anthers are selected for the culture. So you can use acetocarmine for this staining and selecting the uninucleate pollen grain which are present in the anther. Then now we will see what is the pollen culture. So the isolated pollen grains or microspores are inoculated on the MS medium, and which pollen grains or microspore are selected for the culture. Keep in mind, those are uninucleated and those are squeezed from the intact anther. In pollen culture, the anther is not inoculated in the medium. The pollens, isolated pollen or microspore, are squeezed aseptically from the intact anther, and those are cultured on the nutrient medium. And after that, what happens? The pollen grain may develop into embryoids or callus tissue. Those will not produce male gametes, right? In nature, what happens? These microspore and pollen grains divide by mitotically, and that will give rise to two male gametes. But in vitro, the pollen or the microspore that develop into the embryoids and callus tissue, and that will give rise to haploid plantlets through the embryogenesis or organogenesis. That is the pollen culture. And in exam, you will get the question for 2 marks. What is meant by androgenesis? So androgenesis is the in vitro development of the haploid plants from totipotent pollen grains through the series of cell division and differentiation. So, or what are the anther culture or pollen culture? It's one. Keep in mind what is androgenesis. Don't get confused. Then you will ask for what are the 2 different types or methods of androgenesis. Those are the direct and indirect. What is direct or androgenesis? In that, the microspore that will behave like a zygote, that will undergo the change and that will form plantlets. That means there is no callus phase. The microspore directly give rise to the plants. Indirect androgenesis, there is callus phase is involved. So microspore that will divide and form the callus. Then that callus will differentiate into plantlets. So this is same as a somatic embryogenesis or organogenesis. Direct in direct androgenesis, the microspore will give rise to the haploid plantlets. And indirect androgenesis, the microspore first divide and that will form callus. Then callus will differentiate into haploid plantlets. Keep in mind, anther culture and pollen culture are used for the haploid plant production. Then, so this is about the definition. Then for now we will go for introduction. So what happens in nature? The production of haploid plant is very rare phenomenon, and that happens in some species. Those will not happen in all the species. So before this artificial production of plant through the anther or pollen culture, these are the methods. Those are tried. For example, distant hybridization, delayed pollination, then like treatment like temperature shocks or hormone treatment, or irradiation of pollen grains. That means you are going to irradiate this pollen grain with X-ray or UV rays. So these are the methods, but these are the methods are not reliable or not reproducible. So after that, there were 2 scientists. The name of those scientists is Guha and Maheshwari. Keep in mind, those both scientists were the Indian scientists, and they have developed this haploid culture technique for the first time. And the technique involved the production of haploid plant through the anther culture, and they have used the Datura plant for their experiment. After that, there are this anther androgenesis is tried in numerous plant species which are economically important. So now we will see technique for anther culture and for pollen culture. So first we will see anther culture. So you can see in this image, this is flowering plant having the buds. So you can go for the flower buds which are unopened. Then treat this flower plant with the teapot. So that's mean you are going to follow the surface sterilization method. Then the teapot is surface sterilant. Wash these flower buds with 5% of teapot. Then decant that teapot and wash the flower buds, unopened flower buds, with running water, tap water. Then transfer this flower bud inside the laminar airflow. Then you can again use these flower buds for surface sterilization. For example, you can use HgCl2. Then you can give this treatment of HgCl2 for 3 minutes. Then after treatment of HgCl2, you can go for the 3 washes of double distilled water. Then you can excise the anther from this flower bud. Then after excision of this anther, you can remove the filament and then transfer this intact anther on petri plate which contains the medium. So this is anther culture technique, and that anther may give rise to the callus. The callus will develop into shoot or root, that is organogenesis, or the anther may give rise to the embryoids. And finally, both the organogenesis or embryogenesis will give rise to haploid plantlet. So you can go through these slides, and you can select the plant having the first flush of flowers or buds. So this is about anther culture. In next lecture we will see pollen culture. So thank you.